Okay, so welcome back to this next video in which we are discussing protein folding and protein folding diseases. Okay, right. So we're in the process of discussing proteostasis, which remember is the maintenance of a normal healthy proteome within a cell. Okay, now we've discussed that this process involves synthesizing new proteins and then helping those nascent polypeptides to fold into the correct conformation. Okay, then if the native protein conformations happen to misfold, we then need to help them refold into the correct native conformation. And finally, if they're misfolded beyond repair, beyond help, then we need to degrade them. Okay, we've now looked at the mechanisms, or at least we've looked at two examples of mechanisms by which proteins can be helped to fold. Okay, so we've looked at the heat shock protein 70 family of chaperones and the chaperonins. What we're now going to turn our attention to is degradation pathways. And the two degradation pathways that we're going to look at, the ubiquitin proteasome pathway and autophagy, both of those are only found in eukaryotes. Okay, so we end this point where we are discussing things in E. coli then. Okay, so let's start with the ubiquitin proteasome pathway. Okay, so I just firstly want to highlight how important protein degradation by these pathways is. Okay, so it's estimated that around a quarter of proteins that are synthesized fail to fold in the correct manner. Okay, so you synthesize the polypeptide, it's a nascent polypeptide, it's estimated that a quarter of them fail to fold and therefore have to actually be sent down degradation pathways. So these pathways are not niche, okay, they're very, very important within cells for maintaining normal proteostasis. Okay, only some of the proteins that you actually make are actually going to fold into the functional correct conformation, and the ones that don't, they need to be degraded basically. So it is a wasteful process, protein synthesis. Okay, so basically this pathway consists of you firstly labeling proteins up for degradation, and then this label sending them for degradation in the proteasome. Okay, so the name gets it across perfectly. It is the ubiquitin proteasome pathway. You label molecules up with this molecule ubiquitin, and that then targets them to the proteasome for destruction. Okay, so let's start with that labeling process. Let's start with how do you attach this ubiquitin molecule onto proteins? Well, firstly, what we need to investigate is what actually is ubiquitin in the first place. Okay, so ubiquitin is actually a polypeptide itself. It's a protein. Okay, it's a short protein. A protein of, well, I don't even need approximately, it is equal to 76 amino acids. Okay, so it's a nice short protein. Okay, and it can be attached onto other proteins via its terminal amino acid. Okay, so let me draw the ubiquitin protein here. Okay, so here is its amino terminus, and I'll represent it in this nascent structure that I'm drawing here, but in reality it will have a folded structure. I'm just representing it like this for simplicity. Okay, then here is its C-terminus over here. Now the final amino acid, if you're interested, is actually a glycine residue. So this is a very simple amino acid where the R group is merely a hydrogen atom. Okay, now, what other preliminary remarks can I make on ubiquitin? Only that we will colour it in green, I think, and also that it's often abbreviated down to UB for ubiquitin. Okay, oh, and another comment I can make is that it is called ubiquitin because it is ubiquitous. You find it in all cells of the body. You find it not just in all cells of the human body, you find it in all eukaryotic cells, and it's pretty much identical across all eukaryotic cells. Okay, so this is a very, very conserved protein. Nature has found this, it works, and it has run with it, okay, and it hasn't really changed it that much at all. Okay, so that's why it's called ubiquitin, because of how ubiquitous this protein is. Okay, so basically the labeling process isn't as simple as you might think. Okay, it would be really nice and simple if literally there was just an enzyme which grabbed a ubiquitin molecule and reacted it with a polypeptide and then it was stuck on. Okay, that is eventually going to happen right at the end. The problem is you don't actually just grab a free ubiquitin molecule from the cytoplasm and attach it onto a polypeptide. Firstly, what has to happen is the ubiquitin has to be loaded onto a bunch of other enzymes first. So we have to go through this process first. So let me describe this initial part of the ubiquitin proteasome pathway. So it starts off then with enzymes that are for short called E1 enzymes. Okay, but E1 enzymes have a longer name. Their longer name is to call them ubiquitin activating enzymes. Okay, and these are the first enzymes which are actually going to bind to ubiquitin. Okay, so you can either call these E1 enzymes or ubiquitin activating enzymes. Either name is perfectly reasonable and people will know what you mean. Okay, now, there is not just one E1 enzyme in humans, and that's going to be true of all the other enzymes involved in this pathway. We're also going to see E2 enzymes and E3 enzymes later on, okay? And for all of them, they are generic names, okay? There are absolutely loads of the things in humans. There's loads of E1 enzymes, there's loads of E2 enzymes, and there's loads of E3 enzymes. They all do the same thing, so we're using the generic name of ubiquitin activating enzyme, but in reality there is not just one ubiquitin activating enzyme, there are loads of them. Okay, so this is an E1 enzyme, or a ubiquitin activating enzyme, whichever name you want to use. Okay, right, so all that we actually need to know about the structure of the E1 activating enzyme is that it has a file group. Okay, so the rest of the structure is being beautifully represented by this turquoise box, and the only bit that we actually need to actually draw out as a molecular formula is this file group here. Okay, so we've seen file groups before. They were one of the functional groups in a cysteine residue. Okay, they have similar chemical properties to alcohol groups. Okay, because sulfur is actually in the same group of the periodic table as oxygen, and therefore it has similar chemical properties to oxygen. And of course, if that was an oxygen there, that would just be an alcohol group. Okay, so if you're not familiar with the chemistry of file groups, I would encourage you just to think of them as being like alcohol groups. Okay, now, as far as we're going to see, they're going to behave very like alcohol groups because they are going to react with the carboxylic acid terminus of the ubiquitin protein, basically, okay, in exactly the same way as an alcohol group would. So alcohol groups can react with carboxylic acid groups in the famous esterification reaction. Okay, well, file groups can react with carboxylic acid groups in pretty much an identical reaction, but instead of forming ester links, what you form is what's known as a thioester link. Okay, so let me show this. So basically what you're going to do is, if I draw the carboxylic acid group here, like so, and then the rest of the ubiquitin is here, and I might draw it folded up now. Okay, there we go, just to be inconsistent, but there's the ubiquitin protein here. Okay, in green. Right, the colour coding will at least be consistent. Okay, and now what we're going to do to form a thioester link is we're going to take off the alcohol group from the carboxylic acid group. We're going to take off the hydrogen from the file group, okay, just like we would in an esterification reaction. We're going to combine those two things together to make water, so it's going to be a condensation reaction because it's going to produce water. Okay, and then we're going to bind the sulfur atom of the file group to the carbon atom of the carboxylic acid group. And what we'll overall end up with now is our E1 enzyme here, like so, with our file group sticking off the side, now with this thioester link between the file group and the carboxylic acid group. And here's our ubiquitin here. And there's the amino group of the ubiquitin. So let's colour code everything in. So here in turquoise is our E1 enzyme, or ubiquitin not conjugating enzyme. We'll see ubiquitin conjugating enzymes. It's a ubiquitin activating enzyme. Okay, and then here in green, this is our ubiquitin over there. Okay, so we've now loaded the ubiquitin onto this enzyme. So as I say, we're getting the ubiquitin ready to be transferred onto our protein of choice. Okay, and I might just draw out the protein of choice in a moment. Well, right now actually. So here, this protein over here, which will colour in I think red, this is the protein that we are aiming to destroy, just so that we can see where this is going. This is the protein that we want to stick that ubiquitin molecule onto. Okay, we want to tag this protein for destruction. Okay, so we might call it the condemned protein over here. Okay, so this is what we're aiming to attach the ubiquitin group onto, but we're not there yet. We have to attach it onto a bunch of things in between before we finally transfer it onto the condemned protein. Okay, now it gets worse, unfortunately. You might think, well, can't we just transfer it off the E1 onto the condemned protein? Well, before we can do that, it has to be transferred onto another protein. We can't, basically, is the answer. We can't transfer it off the E1 onto the condemned protein. In fact, we transferred the ubiquitin onto the E1 merely so that we can now transfer it onto another protein, which then we will transfer it off onto the condemned protein. Okay, so the next enzyme in this cascade then is an E2 enzyme. Ooh, and I've missed something important. Okay, I've missed the fact that this first reaction is dependent on ATP hydrolysis. So for this first reaction to occur, you have to hydrolyze ATP, and it's an odd hydrolysis of ATP. You don't produce adenosine diphosphate, you actually produce adenosine monophosphate and a pyrophosphate molecule, which is a weird way to hydrolyze ATP, but that's apparently the way it's done in this reaction here. Okay, right, so that's the only reaction in this entire pathway that's, the only reaction at this initial labelling stage that is going to be ATP dependent. Okay, right, so what's now going to happen is we're going to transfer the ubiquitin group from the E1 enzyme onto an enzyme that's now going to be an E2 enzyme. And again, the only thing that's important for us to actually know about the E2 enzyme is that it has a file group. Okay, so we'll colour in this E2 enzyme here in blue. Okay, and it has this file group here. Okay, so just like E1 enzymes had this other name, they were all so-called ubiquitin activating enzymes, the E2 enzymes have another name. They are also known as the ubiquitin conjugating enzymes. So you can either call this an E2 enzyme or you can call it a ubiquitin conjugating enzyme. Okay, and the same thing holds true in the case of the ubiquitin conjugating enzymes as for what I said with the ubiquitin activating enzymes, in that there are loads of them. Okay, so there is not just one E2 enzyme, there are loads of them that can all perform the same function. Okay, so it's a whole family of enzymes. So we'll just use the generic name and call it an E2 enzyme or a ubiquitin conjugating enzyme. Okay, so what we're going to do is we're going to transfer the ubiquitin molecule here from the E1 enzyme onto the E2 enzyme, and we're going to transfer the hydrogen of this file group of the E2 enzyme onto the sulfur atom of the E1 enzyme. Okay, so after this reaction, what we will be left over with is now an E1 enzyme here, like so, with a restored file group. So we've regenerated our ubiquitin activating enzyme here, okay, and that will now go off. And now what we're going to have is an E2 enzyme which now has the ubiquitin attached onto it. So here is the E2 enzyme, and it will have our ubiquitin molecule attached onto it, again via this thioester link here. Okay, like so. So here's the amino terminus. Right, so let me colour everything in again. So here is our E2 enzyme in blue, and then once again we'll colour in the ubiquitin molecule in green. Now you might be thinking, well, does this go on forever? Do they continue E3, E4, E5, E6, just transferring them on? No, we have reached the end. Okay, the next enzyme is actually going to transfer the ubiquitin protein onto the condemned protein. So, we now need to discuss what residue of the condemned protein is the ubiquitin going to be added onto. Well, the answer as far as which amino acid residue is easy. It's going to be a lysine residue, okay? Now, I'll just show you the structure of a lysine residue, just in case you don't know. Lysine residues have an amino group right at the end of their R group. So let me just show this. So here is the core amino acid structure. Okay, so the amino group here, and again I'm drawing a residue. So the amino group as though it's bound to the carboxylic acid group of the amino acid before it. The alpha carbon here with a hydrogen coming off, and the carboxylic acid group again bound to the amino group of the amino acid behind it. And then the R group of lysine has 4 methylene groups. Now, because I can't be bothered to draw 4 methylene groups, what I'm going to do is instead draw 1 methylene group, put brackets around that methylene group, and then subscript it 4. That's a useful trick for getting out of having to draw 4 methylene groups. And then finally, the important thing to us is that we have this amino group here. Okay, and it's this amino group that we're going to use to attach the ubiquitin molecule on. Okay, so again, remembering back to your chemistry training, carboxylic acid groups can react with amino groups. Okay, that's called an amide bond, and that's indeed what's going to happen here. You're going to form an amide bond between the carboxylic acid group of the ubiquitin molecule here and the amino group here. Okay, so let me just show this then. So all I'll show now is just the amino group here of the lysine to get me out of having to draw the whole lysine again. And now the ubiquitin molecule is going to be attached on like so. So here's the ubiquitin molecule. So if you can just imagine transferring that over to here, that's what we've got. Okay, and then of course the hydrogen that we had to take off to do that will then end up back on that sulfur atom of the E2 ubiquitin conjugating enzyme. Okay, and everything will be happy. Right, so that's simple. We're going to add it onto a lysine residue, but which lysine residue? Are we going to add them onto all lysine residues within all proteins? No, of course not. We're going to add them onto specific lysine residues on specific proteins, but the actual characteristic that you have to have in order to end up getting ubiquitin molecules attached onto your lysine residues is not understood at all, really. Okay, so we don't understand how cells decide which lysine residues are going to get ubiquitin molecules attached onto them. Okay, and we don't really have a good understanding as to how even the machinery decides which proteins to attach ubiquitin molecules onto, never mind which lysines within the protein it decides to attach them onto. Okay, so that's an area where, you know, we'll find out more in years to come. Okay, so basically there's one final enzyme that I need to mention, which is the enzyme that actually catalyzes the transferring of the ubiquitin molecule from the E2 enzyme onto the lysine residue within the condemned protein. And I might just actually put in my condemned protein here. So this is my condemned protein, which, remember, we agreed we were going to show in red. Okay, so here in red, here is the condemned protein, like so. And we will have some lysine residue here. Now the single letter code for a lysine residue is K, so I'll just denote that as a K residue there. Okay, so what is the enzyme that actually catalyzes this transferring of the ubiquitin molecule from the E2 enzyme onto our condemned protein? Well, it's known as an E3 enzyme, and I'm actually going to draw this in the classical way to draw an enzyme, okay, with an active site there. So this is our E3 enzyme, and again there is another name for E3 enzymes. Okay, they are also known as ubiquitin ligase enzymes. Okay, so this is also called a ubiquitin ligase. And again, the same holds true as held true for E1 and E2, in that there are absolutely loads of different ubiquitin ligase enzymes within eukaryotic cells. Okay, so there is not just one ubiquitin ligase, there are loads of them. And I think actually the vastness of how many there are gets bigger as you go down. So there's a few E1s, there's slightly more E2s, and then there's absolutely a huge number of E3s, which probably represents the fact that these have to be hugely diverse because they're going to be adding these ubiquitin molecules onto a hugely diverse range of different proteins. Okay, right. So the next thing that I need to tell you about is that that isn't really enough to get this protein destroyed by the proteasome. Okay, one miserable little ubiquitin molecule isn't a big enough label. Instead, what you have to do is actually attach on multiple ubiquitin groups. Now you might think, well, are we going to attach them onto all the lysines of this polypeptide? Well, no is the answer. What we do is we add more ubiquitin molecules onto lysines within the ubiquitin molecule that we have just added onto our condemned protein. Okay, so specifically in the ubiquitin molecule here, there is a lysine at position 48. Okay, what we can do is we can add more ubiquitin molecules onto that. Okay, so here comes another ubiquitin molecule. Okay, so I'll show the amino group of this lysine here, and then what you'll have is you'll have the carboxylic acid group of another ubiquitin protein, which I'll show here. Okay, and then here's the amino group of this other ubiquitin protein here. And then of course it doesn't end there. This new ubiquitin protein that we've just attached on here, this will also have a lysine 48. Okay, so here is K48 of this one, and we can attach another ubiquitin molecule onto that, and so on and so on and so on, until we build a massive great chain of these ubiquitin molecules all attached onto one another, and the name for that is a polyubiquitin chain, basically. So this is a polyubiquitin chain. Okay, so if you want your protein to actually be destroyed by the proteasome, what you need to do is attach on a polyubiquitin chain, or a polyubiquitin tail, some people often call it, okay, onto your condemned protein, and that then is enough to actually get it noticed by the proteasome. Okay, so let's now actually discuss the proteasome itself. Okay, now this is quite a terrifying thing in the world of molecular biology. Okay, this is something that proteins go into and fragments of proteins come out of. Okay, so let me show you then the structure of the proteasome. So the proteasome is often called the 26S proteasome. Okay, which refers to Shredberg units of the speed at which it has to be spun to centrifuge things out, okay, to pellet things out. It does reflect the size of the structure to an extent, okay, but if you work on that basis, it becomes very complicated to explain other things. Okay, so this does represent roughly how big it is, but when you split the proteasome up into two different bits and then work out what their Shredberg units are, you don't get two numbers that add up to this one, okay, so it doesn't work additively, okay, and that'll become clearer in a moment. Okay, so the 26S proteasome then, it is made up out of a core portion that is 20S, okay, and it's made out of added on to the 20S core, you've also got two 19S regulatory particles, and that's what I mean by them not adding up to give the 26. Okay, so it doesn't add up as far as size goes. Okay, but it is roughly related to how big the protein is. Okay, right. It's really how dense the protein is rather than how big it is. Okay, so let me firstly draw the 20S core here. Okay, like so. The 20S core is effectively a tube, basically, made up of a huge number of proteins, and that's probably one of the first things that I should have said rather than faffing about with the Shredberg units. Proteasomes are not just one protein, okay? They are a massive, massive, massive complex of loads of proteins that are all different, that have been stuck together to perform this incredible role, okay? So we're starting off with this 20S core portion of the proteasome, which itself is made up out of 28 different proteins stuck together, okay? And these 28 different proteins are arranged in 4 rings of 7. Okay, so let me show this. So here's the first ring, here's the second ring, and here's the third ring. So we're seeing heptamers once again. So here's the entrance to the 20S core, and these rings are all going to be made up of 7 proteins. 1, 2, 3, 4, 5, 6, 7. Okay, and I'll divide it up along here as well, like so. So those are the 4 rings, each of 7 proteins. Okay, now these rings closest to the center, which I'm colouring in orange here, these are known as the inner rings, okay, consisting of 7 separate proteins. Okay, so I'll just label that up. So these ones in the center, these are the inner rings. Okay, and each of them obviously consists of 7 separate subunits, and these 7 separate subunits are known as beta subunits. Okay, so you take 7 beta subunits, join them together in a heptameric ring, and that makes the inner rings. And you take 2 of those inner rings, and that makes the center of the 20S core of the proteasome. Okay, now, these beta subunits are not identical, okay, so they're all different basically, and many of the beta subunits have protease activity, and it is these beta subunits that are going to be cutting up the polypeptide as it moves through this tunnel basically, as it moves through the 20S core. Okay, then we have the 2 outer rings, which I'll colour in green here. So here's one outer ring on the left here, and then we have another outer ring here, which again are these heptameric rings, so each one of them has 7 separate subunits in, and these are made out of alpha subunits rather than beta subunits. So these are the outer rings, and they're alpha subunits, and those are not going to have the protease activity that the beta subunits have. Okay, right, so that's the structure of the 20S core of a proteasome. Okay, and this is the portion that the polypeptide is going to be threaded through, okay, and as the polypeptide is threaded through there, the proteases that make up the walls of the 20S core are going to be degrading the peptide bonds and breaking it up into little fragments. Okay, right, now, on either side then of the 20S core, you have extra portions, one here and the other one here, which I'll colour in blue. Okay, so here's one in blue here, here's the other in blue here. Okay, these are what are known as 19S regulatory subunits, okay, and now hopefully it becomes completely clear what I meant by them not adding up. Okay, so you don't add 19 to 19 to 20 to get 26. Okay, so the Schredberg units don't add up. So the entire proteasome is the 26S proteasome, but it's not got the Schredberg units that you'd expect if you were just adding up the Schredberg units of the 3 individual components. Okay, so this is the 19S regulatory particle. Okay, now, it is the 19S regulatory particle that is going to be involved in binding to the polyubiquitinated chain of the condemned protein. Okay, so if we've got our condemned protein here, which has got this polyubiquitin chain here, what's going to happen is the polyubiquitin chain is going to bind to the 19S regulatory particle. Okay, and then the 19S regulatory particle is going to feed the condemned protein into the tunnel of the 20S core, and then it's going to be broken down into small fragments of amino acids ranging between sizes of 3 and 25 amino acids. Meanwhile, it is the 19S regulatory particle that will break down the polyubiquitin chain. Okay, right. So that then is the ubiquitin proteasome pathway. That's how we label proteins up with these polyubiquitin chains, which sends them to the proteasome, where they're degraded into small fragments of amino acids.